Adaptive immunity. This is your tertiary source of host defense and is called into action. And the link that we've been talking about is via macrophages and dendritic cells. It's cell-mediated and humoral immunity. And we'll talk more about what we mean by these two very, very shortly. Problem with the adaptive response is it takes time to mount this defense. Why? Because macrophages come in a little bit later after acute inflammation begins, and as a result, when it gets into the scene, it has to basically start their process. So they're going to engulf the pathogen, and we'll see how this plays out very, very shortly. Not only that, but it has to get the mediators going to attract these cells to the specific scene. And we'll see how this plays out through basically a series of videos. And these videos are all posted for you on Quercus, so make sure you go for them. The responses to the adaptive response are really antigen specific. Thank goodness. And now when we're comparing it to, say, acute inflammation in the innate response, those are very generic responses. It's an overall global response to that pathogen. But now we're getting a response that is antigen specific. And we're dealing with T cells and B cells that are specific for a specific type of antigen. And they form memory B cells and memory T cells, which allows for a secondary response to that antigen to be much quicker. And we'll see a bit of that later on as well. And that's why it remembers the specific pathogens via these memory T cells and memory B cells. It's quite the response. Let's look at the antigens as well as the antibodies. So an antigen, like we talked about earlier, is really that etiological agent that we've been dealing with. It's a foreign body. And we can take this antigen and cut it up. We cut it up into what we call a little epitope. And this is the region that reacts with an antibody, the T cell receptor, as we will see very shortly, as well as the B cell receptors. And we'll see that very shortly as well. Antibodies are what's produced from plasma cells. So B cells, once they become stimulated, they'll differentiate into plasma cells. And they secrete antibodies that are basically marking antigens for elimination. And we have types of immunoglobulins, which are what produce via the plasma cells. So IgM is one of 5, and it stops infections by bacterial toxins as well as viruses. IgG, really one of the prime functions is to protect newborns. IgA is really defense of mucosal surfaces, so the mucous membranes, you see quite a bit there. IgE, many of you probably dread this one, is really involved with allergies, and we'll see how this plays out later on. And IgD really triggers B lymphocytes. It's one of the major functions. So let's look at how all of this happens. And one of the first processes that happens is antigen presentation. And this is fundamental immunology. We're literally taking an antigen as it gets into the cell, usually within a phagosome, it merges with, say, a lysosome, as well as basically the enzymes within that lysosome really start to degrade the actual antigen. And that's the goal, is to take this antigen, break it up into little pieces, take some of these fragments, goes into the ER, goes onto an MHC molecule, as we see here, a little more processing through the Golgi, and then is expressed on the cell surface for the immunology cells, specifically the lymphocytes, specifically T cells, to look at. And the major type of T cell will be the T helper cell. More on that in a minute. But now we're going to show you a couple videos showing you antigen presentation and what MHC molecules are all about.
Special antigen-presenting cells, including monocytes, macrophages, and their relatives, engulf invading pathogens and digest them, producing fragments with antigenic determinants. Vesicles containing MHC proteins fuse with vesicles containing digested pathogens. The antigenic determinants from the pathogens are loaded onto the MHC proteins. MHC proteins are used to present peptides on the surface of the cell. Since the peptides are produced from proteins inside the cell, they allow the cells of the immune system to detect proteins inside the cell without entering the cell. Cells in the body routinely break down proteins into small peptides, which are about 8 to 10 amino acids in length. These peptides enter the endoplasmic reticulum and are loaded onto MHC proteins. The vesicles containing the MHC peptide complexes bud off of the endoplasmic reticulum and are released into the cytosol. Vesicles containing the MHC peptide complexes travel through the cytosol to the cytoplasmic membrane, delivering the complexes to the exterior of the cell. If this cell is functioning normally, the immune system cells will simply ignore this presentation. However, if the cell is infected and thus presenting viral proteins, the immune system cells can notice the problem and take action. Antigen-presenting cells, such as macrophages and dendritic cells, have two classes of special proteins that are used to present antigens on their surfaces. Collectively, these proteins are called MHC, which stands for Major Histocompatibility Complex. Class I MHC proteins are used to present antigens produced within the cell. A different class of MHC proteins, Class II MHC proteins, are used to present antigens engulfed by antigen-presenting cells. Most of the other cells in the body, including infected cells, are also able to present antigens. However, these cells only have the Class I MHC proteins.
Before we proceed to the next videos, I want to just remind you of a couple of things. One, MHC1s are produced on all cell types within our body. MHC2s are produced only on the dendritics and macrophage cells, and those basically are your professional APCs. They attract different cell types. Remember, MHC2 goes for CD4 cells, CD4 plus cells. Those are the helper T cells. And the MHC1s attract CD8 cells, CD8 plus cells. And those are the cytotoxic T cells. Both have very different roles, and we'll see how they play out very, very shortly. So don't forget the types of cells that are involved here. We're dealing with macrophages and dendritic cells. This engulfs these foreign bodies. This sets up the stage for basically for that fundamental immunology to happen. So we're talking about taking pieces of that antigen as an epitope, express that on an MHC molecule, in this particular case, MHC2 molecules, and getting our immune system involved. And we have helper T cells, which basically helps boost the immune response, and cytotoxic T cells, which eliminates infected or neoplastic cells. And we'll see how that plays out too. Don't forget, we have B cells too, and they basically differentiate into plasma cells, and they produce antibodies. And we'll see how that plays out very, very shortly. So let's deal with cell-mediated. And we have two major types. We have the helper T cells, which based on interleukins, and we have the cytotoxic T cells, which really is truly amazing from how they function. They have a porphyrin system, and we'll see how that plays out within videos.
Each helper T cell produces a unique T cell receptor, or TCR, which corresponds to a specific antigen. Each helper T cell also displays a receptor called CD4 on its surface, which recognizes the MHC molecules of antigen-presenting cells. When the helper T cell encounters an antigen-presenting cell with that antigen displayed in an MHC molecule, it signals the antigen-presenting cell to release interleukin-1. The IL-1 stimulates the helper T cell to produce IL-2, as well as IL-2 receptors. The helper T cell can then auto-stimulate itself, keeping it active as it proliferates. Once a helper T cell becomes activated, it proliferates, forming a clone of cells specific to the same antigen. Some of these helper T cells differentiate into long-lived memory T cells, but most mature to produce cytokines to help with the immune response. Helper T cells differentiate into two subpopulations called Th1 and Th2. Th1 cells produce cytokines that primarily activate the cells of cell-mediated immunity, such as macrophages, cytotoxic T cells, and natural killer cells. Th2 cells produce cytokines that activate the B cells involved in humoral immunity. Each cytotoxic T cell produces a unique T cell receptor, which corresponds to a specific antigen. Each cytotoxic T cell also produces a receptor called CD8, which recognizes the MHC molecules of infected cells. When a cytotoxic T cell encounters an infected cell with a specific antigen bound in an MHC molecule, the cytotoxic T cell produces receptors for interleukin-2 and other cytokines such as gamma interferon, IFN-gamma. The cytotoxic T cell can then be activated by cytokines from activated helper T cells. Once a cytotoxic T cell becomes activated, it proliferates, forming a clone of cells specific to the same antigen. Some of these cytotoxic T cells differentiate into long-lived memory T cells, but most mature to attack and destroy infected cells. Cytotoxic T cells can destroy infected cells through multiple pathways. The best understood of these pathways is the perforin granzyme cytotoxic pathway. When the cytotoxic T cell encounters an infected cell with a target antigen bound in an MHC molecule, the T cell secretes two types of protein molecules, perforin and granzyme. The perforin forms a pore in the membrane of the target cell, allowing granzyme to enter the cell. The granzyme activates the enzymes inside the cell, leading to programmed cell death or apoptosis.
Let me ask you a question. Now you understand why cytotoxic T cells are toxic, because they literally can kill your cells. What happens when you start killing your cells? You now have a local site of injury. Guess what happens? You got it. You have now a local acute inflammatory response that can take place within that specific area. So now you get into another loop. Well, that's pretty interesting. See, these are connections that we're making. Okay, so you're finding a connection not only with immunology, but with the innate response. So here we have a response of cytotoxic T cells creating a local tissue injury, and the response to that will be a local inflammatory reaction. Now we're going to get into and talk more about how antibodies come into play. And it's really interesting the way the B cells really are an effector from that perspective and how they actually perform the humoral response too. So we'll see how this plays out with B cells in both what we call T cell independent as well as dependent pathways. Let's begin.
The process of B cell activation is called clonal selection. During this response, a B cell with a specific antigen receptor recognizes an antigen. This cell proliferates to produce a multitude of cells, also called a clone of cells, with the same specificity, which differentiate into memory B cells and plasma cells. This process is called clonal expansion. The antibodies produced by the plasma cells bind to antigens to produce the humoral response. In the simplest form of B-cell activation, an antigen called a T-independent antigen enters the lymphatic tissue and binds to a B-cell receptor, or BCR, on the surface of the B-cell. The B-cell receptors on a particular B-cell are identical and very specific to just one antigen. Once the BCR binds to the T-independent antigen, the B-cell becomes activated. It's now ready to proliferate. However, most antigens are not potent enough to activate B-cells by themselves. In these cases, the B-cell will require a second opinion from a helper T-cell in order to become activated. We call these antigens T-dependent antigens. The helper T-cell recognizes the antigen as it's presented by the B-cell and secretes cytokines to tell the B-cell to become activated. Once a B-cell becomes activated, it proliferates or divides, forming a clone of cells specific to the same antigen. While some of these B-cells differentiate into long-lived memory B-cells, most differentiate into plasma cells, which secrete antibodies against the antigen that activated them. The antibodies can enter the bloodstream and travel throughout the body. Plasma cells only live for a few days, but each one can produce about 2,000 antibodies every second, resulting in enormous quantities of circulating antibodies. The antibodies produced by plasma cells have several functions. Since each antibody has at least 2 antigen binding sites, antibodies can bind to more than one microbial cell, resulting in agglutination or clumping. This agglutination helps remove the microorganisms from the body. Antibodies can also neutralize toxins, viruses, and bacteria by binding to critical portions of the toxin or blocking proteins necessary for attachment to cells. Antibodies also act as opsonins for phagocytosis and activate the complement system.
The last section of the immune system we'll look at briefly immune system disorders. That includes autoimmunity, which really is your immune system attacking self, and we alluded to that a little earlier. We'll cover that in a bit. Hypersensitivity reactions, where your immune system overreacts, and there is some immunodeficiencies, where your immune system underreacts. So in terms of autoimmunity, I don't want to spend a lot of time on this because it's really such a huge, huge field. And I'm going to just briefly go down, and not going to ignore this particular point, but it does deserve some comments. And this came out of a student that was in one of my 4th year seminar series courses who was working in an immunology lab and specifically dealing with autoimmunity. And his supervisor from the Department of Immunology said this to him, and he then told me about it. And so we call it an unpublished theory, but it can't be overlooked type thing. In fact, somehow you'll have to generate a hypothesis, and the hypothesis must be a testable hypothesis, and that hypothesis also must fit the current data that's out there. And until that happens, I'm dealing with a large degree of skepticism. So T cells are bored. They have to go do something. And this does show me that, it's still a field in its infancy. There's many, many theories as to autoimmunity. And we just touched briefly on how basically these T cells, when they're educated, can, if they bind too tight and they do survive, that they could lead to autoimmunity. And so the ethology is really not fully understood. It can be tissue or organ specific and sometimes non-specific as well. And there's 80 plus diseases identified, and there's probably more coming down the pipe every X number of years. And so it's such a huge, huge field. Some of it includes arthritis. It's debatable whether or not Crohn's disease is partially autoimmune, or it can be partially via your innate response, and there's also a very large genetic component, and so dealing with mutations, etc., etc. So have a look at this, especially this one, and to see how many of these facets are involved with this specific disease. I mean, Crohn's disease, by the way, is a type of inflammatory bowel disease, and it's really not very pleasant at all if you have it. Hypersensitivity reactions are really reactions that occur as your immune system hyperreacts, so it overreacts. And there's 4 recognized, and there's probably around 6 or 7 or more that have been defined. And the 4, and we're just going to have a look at 2 of them, is immediate, which involves allergies, cytotoxic, immune complex, and delayed. And we see this one normally within tuberculosis, and we'll have a look at that in a moment. So in terms of immediate hypersensitivity, and there's a normal hypersensitivity, if you would, as well as an extreme version. Extreme version leads to what we call anaphylactic shock, which is really not good. But what everybody has in terms of allergies, which is not anaphylaxia, basically involves exposure, and most of these hypersensitivity reactions will always be on exposure to the antigen for a second time. So that's key for these hypersensitivity reactions. So we have our antigen as a second exposure to give us essentially the reaction mediated mechanism. So here we go. And so we have our APC interact with the antigen. You're making B cells and T cells, etc., etc. In addition, upon making B cells, we start to make this immunoglobulin, IgE. IgE has specifically mast cell binding sites. They go onto and load onto the mast cells. Where do you find mast cells? Mast cells can be found especially in connective tissue. They're found within that region there, and they are found especially in higher numbers surrounding capillaries. Not always. You'll see them throughout the actual connective tissue, but they do have increased numbers around capillaries within that region, within basically what we call the mucosa membranes, the mucous membranes. And on subsequent exposure, so we have now the same antigen coming across our table again, it elicits a very vast response. And we have huge releases of all these mediators of acute inflammation. And we see histamines, what do? Well, they cause leakage of blood vessels. And we see that happening in acute inflammation. Well, here, you're really causing those symptoms. You're releasing these histamines. You're creating all this mucus all of a sudden that comes out into your nasal cavities, etc., etc. And it gives you these symptoms. This is just another diagram showing you exactly the same thing, all the way down to these symptoms because of these chemical mediators, because of the second response to that antigen via the mast cell. So let's have a look at a video to explain that in further detail.
Some people develop an allergic reaction or hypersensitivity when exposed to substances such as dust, pollens, animal dander, or penicillin. This hypersensitivity is mediated by IgE. Sensitization occurs when the antigen makes contact with some part of the body. The antigen is taken up, processed by antigen-presenting cells, and presented on a class II MHC to T helper cells. Tissues under the mucous membranes are rich in B cells committed to IgE production, and IgE-producing cells are more abundant in persons susceptible to allergies. The T helper cells produce cytokines which stimulate these B cells to proliferate and differentiate into IgE-producing plasma cells. As IgE is produced in specific areas of the body, the IgE molecules attach via their constant regions to receptors on nearby mast cells. Mast cells contain granules packed with chemicals that induce a hypersensitivity response. Once attached, the IgE molecules can survive for many weeks. The individual is now sensitized to the antigen. When exposed to the antigen for a second time, the antigen binds to the IgE antibodies on the mast cells. To trigger a response, two cell-bound IgE molecules must react with a specific antigen. Within seconds of the reaction, the mast cell releases histamine and other mediators of the inflammatory response from the granules, triggering a variety of symptoms.
Question for you. What do you take to alleviate these symptoms? You got it. Antihistamines. And they work actually quite well in terms of alleviating some of these symptoms, especially mucus secretion, because it affects the actual capillary dilation. Perfect. Good. Now we're going to get into another type of hypersensitivity reaction, that is delayed type hypersensitivity. And this involves basically T cells, specifically T helper 1 cells. And the easiest thing to do is to go to the video for the explanation of how this works. Now, what I want you to do is to think about, in this particular example, it's contact dermatitis, but think how this would be in relationship to tuberculosis. Try and do a little investigation of that, and we'll address those questions when we get to that specific lecture. Here we go.
Harmful effects produced by the mechanisms of cell-mediated immunity are referred to as delayed hypersensitivity. Delayed hypersensitivity is mediated by Th1 cells, also called TD cells, and is responsible for contact dermatitis, tissue damage in a variety of infectious diseases, and rejection of tissue grafts. A contact hypersensitivity, such as poison oak dermatitis, is initiated by contact with the antigen, which is also called the allergen. During contact, a small molecule from the plant, called a hapten, binds to a carrier protein in the host. The carrier protein with bound hapten is ingested by a macrophage, and the hapten peptide is then presented on the surface of the macrophage on a class II MHC. Th1 cells with T-cell receptors capable of recognizing the hapten peptide antigen interact with the presented antigen, become activated, and increase in numbers. When the host comes in contact with the allergen a second time, the Th1 cells react with the hapten peptide being presented on the macrophage and release cytokines, resulting in attraction of more macrophages, followed by inflammation and skin lesions. Characteristic skin lesions appear after 24 hours, reaching their peak at 48 to 72 hours after the second exposure to the plant.
The next lecture will be human microbe interactions. So if you can read your textbook if you wish, it's completely voluntary, chapters 6 and 10, and gently read 8 and 9. It'll give you a good basis for that specific lecture. And this will be the last lecture before the actual midterm exam. So the midterm exam will be everything up to including human microbe interactions.